Hey guys, welcome to our first set of notes for chapter 24. We're talking invertebrates this chapter, and this chapter is a big one, and it covers a lot of topics, but it just kind of doesn't cover any of them in too much detail. But just so you know, we're going to cover a lot of ground with this chapter, so here you go. Invertebrates, of course, no duh, they're animals without a backbone. They're the most diverse and the most numerous in land and in the oceans as well. Marine invertebrates are super important, and your book gives several examples of them producing secondary metabolites. Secondary metabolite is something that an animal or plant will produce through its metabolism that might not be the primary goal necessarily for getting energy, but is a secondary part of what they would normally be doing. And those secondary metabolites can help them survive. Some might be poisons, some might be chemicals that keep predators from eating them, anything like that. And so we're just sort of barely getting into what these chemicals could be useful for humans, for medicine, or for treating different diseases, that sort of stuff. And one of the examples of that is the cone snail, and the cone snail I think is in the Pacific Ocean, and it actually preys on fish, and it has this harpoon in the snail, and it will shoot it out and stab a fish with it, and the venom in that harpoon will paralyze the fish because a snail has to bring the whole fish in through its radula and it can't chew them up. And so it can't have a fish flopping around while it's eating it or it'd get injured. And so it paralyzes the fish and it eats it while it's still alive, which is kind of wild. So that same compound that paralyzes the fish, it actually works in humans. And if we use it in really, really, really small doses, we can use it to relieve pain that other painkillers aren't taking care of. So we're testing some other chemicals from cone snail venom, and we're looking at treatments for epilepsy, certain types of cancer, and even diabetes. So it's really an untapped resource that we're just barely getting into. Literally millions of different chemicals that are made by different marine invertebrates. So some other medicines that we got from the sea and from invertebrates is AZT. That's a medicine that we use to treat AIDS. We got that from a sponge. And then we also have gorgonians, which are relatives of sea anemones, and they give us a whole bunch of different anti-inflammatory compounds. And so we get a lot from these animals. So what's it take to be an animal? Animals are multi-celled, so more than one cell, obviously, and then heterotroph means they're not photosynthesizing, they're not getting their energy from inorganic compounds, sunlight, stuff like that. They're eating some other living thing. So they have to ingest food, they have to digest food and absorb it, kind of, no kidding, right? They're going to develop from an embryo into an adult form. And more often than not, I might be tempted to say always, the embryo looks significantly different from the adult. Most of these guys are going to reproduce sexually. Well, so a few of them we're going to talk about have the option for asexual reproduction, and it can kind of depend on what their environment's like, which way they'll choose to do that. And they need to move at some point during their lifetime. Some animals can be fixed in one place or sessile when they're adults, but they can swim around or move around when they're larvae or when they're pupa or when they're a medusa or whatever you want to call their younger life form that can move around. And so here's how you would kind of break down all of our animals. Multicellulars, all of them. Sponges do not have true tissues. They're just a collection of cells. You get true tissues in everything else. Radially symmetrical as adults, that's going to be your cnidarians. And then the rest are going to have bilateral symmetry, which means you can cut them top to bottom, and you get a true left side and a true right side that are essentially mirror images. We do get a little bit of variety there. And then you get protostomes and deuterostomes, which has a lot to do with body cavities and designs internally, and those that molt, like roundworms and arthropods, and those that don't molt, like flatworms and roundworms and mollusks like snails. So that's some ways that we can kind of divide these up. Your embryo structure, we're going to look at kind of, I don't know if I can really say simplest to most complex, but kind of. We're starting with like jellyfish, and we're ending at insects, which are incredibly, like impossibly diverse. So we kind of go simplest to most complex. So jellies and cnidarians have two tissue layers, and that's it. They have the outside layer that's called the ectoderm, and they have an inside layer that's called an endoderm. And there's not a lot of division within them as far as their different types of tissues. They're pretty simple. They're a big hollow body cavity. and they will digest food in the big hollow body cavity, and the food will diffuse across the body, and that's about it. They're pretty simple. Other animals that are a little more complicated have a mesoderm, and if you have a mesoderm and you get organ systems that develop from a mesoderm when you're an embryo, then you can have what's called a coelom, and some animals have coeloms and some animals don't. So it's another way that we can kind of divide these guys out. And our internal organs are going to develop from that mesoderm. And so jellyfish, not much in the way of internal organs. Cnidarians, not much in the way of internal organs. Everything else, starfish, insects, earthworms, they're going to have a pretty wide variety of internal organs because of that mesoderm. And so those are your layers: endo, in, meso, middle, ecto, around or out. And so you can kind of sort them out that way. And here's the picture of the first little bundle of cells that would be, I guess past the zygote maybe, but the first embryo. And as it starts to make folds, we can get an endoderm, most certainly, and an ectoderm, which is the outside. And then in some of them, we get the mesoderm. And the mesoderm, that has the potential, to turn into things like muscle and nerves and glands and organs and all of this stuff that kind of makes animals more complicated. So sponges in the ocean are asymmetrical. We already mentioned that animals could be radially symmetrical, like spokes on a wheel, where they kind of all go around a central axis, or they can be bilaterally symmetrical, which is right and left mirror images if you cut them in half. Asymmetrical, A is without, so without symmetry means sponges don't really have a set shape. They're just kind of whatever shape they want to be, and you can't really cut them any particular way to get symmetrical sides. So jellies and other cnidarians are going to have radial symmetry, which we already talked about. And then if you have a 3-layer body plan, which means you get really cool things like muscles and internal organs, then it's a pretty safe bet that you're going to be bilateral. Even really simple stuff like flatworms and like earthworms, which is basically like a little round tube, earthworms if you're fishing or whatever. Those are bilaterally symmetrical. You can cut those in half just like a human and get left and right mirror images. So as we move up from jellyfish into other invertebrates, like roundworms or flatworms even, we get a process called cephalization. Cephalo means head, and so cephalization means the formation of a head. Jellyfish don't particularly have a head, and sponges don't particularly have a head, but flatworms and roundworms have a big group of nerves that doesn't really quite count as a brain yet, but it's a big group of nerves, so we call it a ganglion, or if it's plural, ganglia, and those are all clustered at the front of the animal, and so we kind of call this the formation of the head. And so cells, basically neurons, sensory structures, eyes, ears, that kind of stuff, become concentrated at the front of the body. And obviously if you have eyes or neurons or the ability to smell or a brain, that's incredibly beneficial for finding food and avoiding predators, where a jellyfish can't do much in the way of avoiding predators or seeing what's around in its environment. They just kind of drift along, and if something touches the tentacles, then they pull it up and eat it. It's a pretty simple way to survive. So here's a couple of confusing words that are certainly new to you, even if you took Gen Bio. It's protostomes and deuterostomes. Protostomes have the first opening on an embryo that's going to become the mouth. And deuterostomes have a mouth that develops from a second embryonic opening. And so it really comes down to how they develop when they're an embryo. Most of your bilateral invertebrates are going to be these protostomes, the very first opening on the embryo. Deuterostomes is going to be some of our invertebrates that are a little more complicated, and all of our vertebrates where the mouth forms from a secondary embryonic opening and from that mesoderm. And so you tend to be a little bit more complicated animal if you're a deuterostome. Digestion, there's lots of ways to digest things, but most certainly everyone's going to need to use enzymes to do it. Sponges, they have little cells in the sponge that just kind of all work together, and they're just going to have a little teeny tiny food particle that they filter out of the ocean, put in the cell, and digest in that big mass of cells in the sponge. Cnidarians and flatworms get a little bit more complicated because they actually have a gastrovascular cavity. Kind of think like our gut, it's not nearly as complicated as humans, where we have like the mouth, the esophagus, stomach, small intestine, blah, blah, blah. But they do have a true gastrovascular cavity. All the food goes into one specific hole. and into one chamber. And so they are quite a bit more complicated than sponges with that. And then when you get to bilateral animals like earthworms, roundworms, us, fish, dogs, cats, they're going to have a complete digestive tract. So generally, two different openings, one for the food to go in, a different opening for the waste to come out. Although that's not always the case. Flatworms have a pharynx, and it'll work for food and waste. But most of the time, you're going to have kind of a one-directional flow from food to waste, which I think we're all thankful for. So here you have the gut cavity in the flatworm, the true gastrovascular cavity, and then he has, the mesoderm in the middle, and that can make the tissues, the organs. He's got some nerves in there and some little teeny tiny kidney-like organs that filter small waste, and then the epidermis, which is the skin. And then you have a very different design with roundworms, even though they don't look that terribly different from the outside. The roundworms have a fluid-filled pseudocoelom, so they don't really quite have a coelom where it's a cavity that's made from the mesoderm and houses other organs. It's not really that way, but it is filled with fluid, so it does give them the shape of a lot of bilaterally symmetrical invertebrates, and they still have a specified gut cavity. So again, much more complicated than a sponge. And then we get to the bilaterally symmetrical annelid. Annelid means segmented worm. So worms have small rings going around them. That's where they get their name, the segmented worm. And they do have a coelom. This right here, this middle layer, if you were to cut them like so, they have this layer inside of them that comes from the mesoderm that houses a whole bunch of different organs and is very diversified, and that's a true coelom. And it has sheets of tissue called mesentery, not that different from the mesentery that suspends the small, large intestine, stomach, pancreas, those sorts of things in a human even, and they're going to hold the gut in place, and that's exactly what they do in you and I as well. They still have an epidermis, which is good because that's the outer skin layer, and they still have a specific gut cavity. They have a mouth, they have a digestive tract, and those are separate from the rest of the body. And so there's a little more complicated design. I guess kind of simplest to most complex, if you want to think about it that way. That would probably be reasonable. So when we look at all these different invertebrates, and from an evolutionary worldview, we have to ask ourselves, how did we get from cells, just being cells living on their own in the ocean, to an animal, which is a collection of cells and tissues? And the most common and probably the most popular idea is what's called the colonial theory. We know that some algae live colonially. They live together in a big ball of algae called like volvox perhaps, and they have the capacity to live alone as a cell, but they almost choose to live together in a big colony. And so maybe the idea is that like we had some cells that were protists that had the option to live alone, but they lived together as one big colony of protists. And eventually they got specialized cell types, where one cell was really good at digestion, but not so good at protecting itself with a tough epithelium, and a different cell had a really strong epithelium, but wasn't so good at digestion. And so they could maybe gradually get to a place where they can't live independently, and they have to live as a colony. And that could give rise to our first simple animal, where we would kind of get those cells to start to differentiate out to a point where you could almost call them tissues. And choanoflagellate cells, which are in sponges, kind of resemble cells in modern sponges, where the idea is these cells have like a flagella that like oscillates and pushes water through, and they get food particles stuck in this weird mucous membrane outside of the cell. And those cells line the entire inside cavity of a sponge, and they get the food, and they move it into the sponge body. And so you can kind of see how this idea would sort of come to be reasonably popular in a naturalistic worldview for that. And so our earliest evidence of animals, we have found some biomarkers in rocks that are dated to about 635 million years old. They are sedimentary rocks, of course, so you understand how those are dated. They're dated by the fossils in the rock. And so animals have been around for a while. 570 million years old, we found fossils of marine invertebrates in Australia, and we have seen dramatic adaptive radiation during what's commonly called the Cambrian explosion. That's about 540 million years ago. All the animal lineages that were present right by the end of that period, which evolutionarily or geologically speaking is like the blink of an eye, and all of these animal body types show up. There's a couple reasons that could potentially be, where evolution was so slow and then evolution was so incredibly fast. One option is the climate warming up as we start to photosynthesize and we make more oxygen and we make more ozone, and the ozone starts to protect us a little bit. Our climate could become a little bit warmer, and also the UV radiation is not nearly as damaging if you want to move up out of that deep ocean water. And the oxygen concentration is increasing because of the photosynthetic cyanobacteria that have been working so hard for so long. And so animals that have the ability to use oxygen as an electron acceptor in the way that they make energy for ATP, those animals can become very, very diverse. And so that's kind of some ideas as to how we can get in our fossil record like nothing, 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 and then boom, here's all these shelled animals and crazy like lobster things that just come out of nowhere. We gotta have an explanation for it. So there's one explanation for it. Some biological factors that might have influenced diversification into all these different species in such a short time is as soon as you have animals, it's pretty likely they want food, and somebody's going to not be a filter feeder, somebody's going to be a predator. And once you have predator-prey interactions, it's going to be a really strong selective pressure to favor those body types that can take the calcium and take the carbon dioxide that's in the ocean and make calcium carbonate, make a hard shell, make a tough exoskeleton. They're going to be harder to eat. And that's going to drive the coevolution between predators and prey to become, whoever is the toughest and has the toughest plates and the best camouflage. We can change animals quickly from there. So that's a pretty good start. We'll stop there at section 4, which is going to be sponges, and we'll jump back into those next time. So thanks for your time.